Good morning students. Welcome to my YouTube channel Mulix Academy. Today's topic is Biochemistry Analyzer. These analyzers are used in our laboratories, and the questions that is very common in exams what are the biochemistry analyzer, what is the automation in laboratory, or differences between semi-auto analyzer and fully automated analyzer. So these questions we will discuss in a lecture. So let's start. See, biochemistry analyzer. What is the mean of analyzer? Analyzer means which will analyze. And what it will analyze? It will analyze our parameters. Like it will analyze sugar, or it will analyze LFT or RFT. So biochemical parameters are analyzed by biochemistry analyzer. Okay, so it is a machine that is based on the principle of photoelectric colorimetric principle. Colorimetric means it measures the color. When there is a reaction between reagent and sample, there a color development, and these analyzer read that color. So it also called colorimetric principle. And photoelectric photometry. Photometry is based on the principle of Beer's law and Lambert's law. We will discuss in another lecture what is Beer's law and what is Lambert's law. For now, we will just remember that these analyzers are based on the principle of photoelectric colorimetric principle. These analyzers are used in hospitals, family planning services stations at all the levels. So these are the uses of these analyzers. See, analyzers are of two types: semi-auto analyzer and fully auto analyzer. Semi means half. Okay, so in these analyzer, LT plus auto analyzer, or LT plus auto analyzer or semi जो machine है, it means LT plus semi auto analyzer will work together. Okay, this is the semi auto analyzer. This is the display जहां पर कि आपका result display होगा. These are the जहां से हम प्रोग्राम कर सकते हैं, जो भी पैरामीटर हमको मेजर करना है उसकी प्रोग्रामिंग. वी कैन डू प्रोग्रामिंग बाय दिस स्टेशन. दिस इज द ट्यूब बाय विच machine can suck the sample or sample plus reagent mixture. Okay, here we use the paper roll and we can print the results. So these are the parts of semi-auto analyzer, and in semi-auto analyzer half function is done by LT or lab technician and half is done by the machine. See, that's why these are called semi-auto analyzer, because the initial stages of sample analysis are performed by lab technician. वो क्या क्या stages हैं? Like pipetting of reagent. See, if I want to analyze the sugar, so what are the steps of mine? Like I will take a test tube. Okay, this is the test tube. I will take reagent in it, glucose reagent. Okay, like 1 ml I am taking the glucose reagent. Then what will I do? I will add the sample. Like 10 microliter I am adding the sugar sample or patient sample, sorry. So this is the test tube. In this is the reagent, and we are adding this. See, this is the sample or patient serum. Okay, then what will happen? This pipetting of reagent is done by lab technician. Pipetting of samples is done by lab technician. Then we will mix it. Reagent and sample, there will be a mixing of reagent and sample, and then incubation. Like if there is incubation of 10 minutes, example, we will incubate it for 10 minutes. Then incubation, mixing and incubation of reaction mixture is done by LT. Okay, LT will keep this mixture in the incubator for 10 minutes or room temperature for 10 minutes. Okay, then there will be a color development. There will be a color development. Now, these all functions are done by LT or lab technician. Reagent pipetting, then sample pipetting, then mixing of reagent and sample, and then incubation of this reaction mixture. Okay, then what will machine do? Then machine will suck the reaction mixture, sorry, and then semi-auto analyzer read this mixture. Maybe this reaction is endpoint or rate of reaction or kinetic reaction. Okay, reaction may be of two types, endpoint or kinetic reaction, or nowadays fixed kinetic also. We will discuss in another lecture what are the types of reaction, what is the endpoint, what is the rate of reaction, what is the fixed kinetic. Maybe the reaction doesn't matter, the reaction is endpoint or rate of reaction. Semi-auto analyzer read it and then display the test result. Okay, result display हो गया. Then this result will print and memorize by the machine, by the semi-auto analyzer. So half function is done by LT and half function is done by the machine. Okay, pipetting of reagent, pipetting of sample, mixing of reaction mixture, and incubation. These steps are done by lab technician, and then reading, then display, then printing, and then memorizing. These are done by the machine. So that is why this is called semi-auto analyzer. Now this is fully automated analyzer. Fully automated analyzer में, like, this is the sample probe, this is the reagent probe. Okay, this is the sample tray, this is the reagent tray. Okay, these are the parts of the fully automated analyzer. Fully means सारे functions जो है वो machine itself खुद से कर रही है. Okay? These analyzer perform all the functions of semi-auto analyzer, and in addition they also perform following functions. Semi-auto analyzer क्या क्या function कर रहा था? जो रिएक्शन मिक्सचर है उसकी जो रिएक्शन है उसको रीड करना, देन रिजल्ट डिस्प्ले करना, देन प्रिंटिंग करना, देन मेमोराइज करना उस रिजल्ट्स को. ये सारे फंक्शन तो फुली ऑटो एनालाइजर करेगा ही, बट इन एडिशन वो क्या करेगा? ऑटोमेटिक डिस्पेंसिंग ऑफ रिएजेंट्स बाय रिएजेंट प्रोब. सेमी ऑटो एनालाइजर में जो रिएजेंट पिपेटिंग बाय द टेक्नीशियन हो रही थी, इन फुली ऑटो एनालाइजर द रिएजेंट डिस्पेंसिंग बाय द रिएजेंट प्रोब. Then the sample dispensing by the sample probe, and then the mixing of reaction mixture is done by the machine only, and then the incubation of reaction mixture is also done by the machine. Okay, there is no need of incubation. अलग से हमें इंक्यूबेटर में रखकर इंक्यूबेट करने की जरूरत नहीं होगी. फुली ऑटोमेटेड एनालाइजर में ही इंक्यूबेशन होगा. Reagent pipetting is done by the machine, sample pipetting is done by the machine, mixing is done by the machine, and the incubation is done by the machine. These all in semi-auto analyzer is done by the lab technician. Okay, so these functions are done by the fully automated analyzer, and साथ ही जो reading, display, result, printing, and then memorization, these all are done by the machine only. So all functions are done by the machine only. So that's why these analyzers are called fully automated analyzer. These analyzers are fully automated from sample addition to result generation. सैंपल ऐड करने से लेकर रिजल्ट जनरेट होने तक के सारे काम जो है वो कौन कर रहा है? सिर्फ मशीन. दैट्स व्हाई दीज एनालाइजर्स आर कॉल्ड फुली ऑटोमेटेड एनालाइजर. The operator only needs to put the sample on the specific position. Like if there is a sample tray, तो ऑपरेटर को क्या करना है? सैंपल ट्रे में सैंपल्स लोड करने हैं, रिएजेंट ट्रे में रिएजेंट लोड करने हैं, एंड देन प्रोग्राम चूज करना है टू स्टार्ट द इंस्ट्रूमेंट. इंस्ट्रूमेंट को स्टार्ट करने के लिए प्रोग्राम चूज करना है और मशीन को स्टार्ट करना है. दिस इज अबाउट द सेमी ऑटो एनालाइजर एंड फुली ऑटोमेटेड एनालाइजर. In the next lecture, we will see the differences between semi-auto analyzer and fully auto analyzer. What are the differences between these two semi and fully auto analyzer? Okay. Thanks for watching.